So Dr. Kate Broderick, she received her Ph.D. from the University of Glasgow in Scotland and conducted her postdoc research at the University of California in San Diego. She joined Inovio in 2006, where she currently leads a diverse research group focused on enhanced delivery techniques for gene-based therapeutics as the Senior Vice President of Preclinical R&D. She's a co-inventor on multiple patents related to DNA vaccine delivery and has served as principal investigator on grants, awards and contracts from leading government agencies and not-for-profits, including Bill and Melinda Gates Foundation, the NIH, the US Department of Defense, Small Business Innovation Research Program, and including a $56 million award from the Coalition for Epidemic Preparedness Innovations, CEPI. In 2018, her particular area of focus is infectious diseases, with an emphasis on emerging targets and those from a biothreat potential. Most recently, Dr. Broderick led the teams that brought the first in human Lassa fever virus vaccine into the clinic, as well as advanced the development of a DNA vaccine for the MERS virus. And she's responsible for driving the development of a DNA vaccine for COVID-19. So Kate, thank you very much indeed for taking the time to speak to us today. And we're going to look forward to hearing your talk, the INO-4800 DNA-based vaccine against COVID-19. Thank you.
Thank you so much for the really kind introduction. It's a real pleasure to be with you today. I think most of you are in Greenwich Mean Time, from what I can tell. Don't be confused by the accent. I've lived now for 20 years in California. Inovio is, for those who may not be familiar with what we do, we are a mid-sized US-based biotech. I have the great pleasure of being at the research site in San Diego, California, alongside our manufacturing facilities. But our headquarters are based in Philadelphia, just outside of Philadelphia on the East Coast. So it's a real pleasure to be here today, and I'm really very, very grateful for the opportunity to speak to you today. I think we're running a little bit over on time, so I can certainly speak very quickly and get us through so that we're still on for the panel discussion. So like many of those of you on the call today, myself and the team here at Inovio have spent the last 15 months really pretty much entirely dedicated to COVID-19, and just wanted to take the opportunity in the next 20 or so slides to kind of share what we've been doing in those 15 months and where we hope to be as we kind of move through the pandemic, and what we see as the future of the pandemic. So again, for those who may not be familiar with our technology, I wanted to kind of start by introducing it at a fairly high level. So our technology platform at Inovio is DNA-based medicines. We have 15 ongoing clinical programs currently in development, and I'll just kind of give you a snapshot of what they are in some later slides. But really, the technology is foundational in the marriage between highly optimized DNA plasmid construct with delivered through our proprietary smart delivery device. And I'll talk about both of those aspects because they're pretty fundamental to what it is that we do. Our vaccines can be either for treatment or prevention, and we target both infectious diseases, which is my particular area of focus, but also oncological targets as well. One of the very nice characteristics of DNA-based medicine is they're generally very well tolerated and induce kind of robust immune responses, and I'll talk a little bit more about that. Because it's DNA, it's a DNA molecule, we don't have any anti-vector responses, and also through kind of enhancements in the formulation, we also don't require any frozen cold chain. So in the concept of a worldwide deployment of vaccines, you can see how that could be very, very beneficial. Although not pertinent to the COVID discussion, but following on from Katie's last talk too, we also have worked pretty extensively in the Ebola fields, Zika and Lassa, just to give some examples. So one of the nice elements of DNA vaccines is that you can co-formulate multiple plasmids within a single vaccine cocktail. So in fact, we've looked at a sort of hemorrhagic fever, where we look at Marburg, Lassa and Ebola all into a single vaccine. And also done the same for mosquito-borne diseases too. So that's one of the nice benefits of DNA, is you can tend to put multiple vaccine constructs together without any issues with competition. So as we think about the situation we're in today, but also kind of then projecting into the future of COVID-19, but also the future of any other pandemic, as I think probably everybody on the call today can recognize that we are still absolutely at a threat for other future pandemics. How does kind of a DNA medicine vaccine fit into that environment? So I've pulled out some of the kind of dimensions of DNA medicine or DNA vaccines that I think really set to position it very well in the context of what we're living through right now, but also any future pandemics, be those flu or disease X. So certainly kind of following very much in the theme of the discussions today, one of the key signatures of DNA vaccines is their ability to produce a really very well defined, robust CD8 response. And as Katie just mentioned, we really believe that T cell responses, of course, are way less susceptible to mutational changes that we're seeing with the variants of concern. And I'll talk a little bit more about how that plays into our kind of fight for against the variants that are circulating at the moment. As is, of course, absolutely key, our vaccines don't just generate T cell responses, but also, of course, the all-important binding and neutralizing antibodies. And I can give you some examples of what we've generated from our phase 2 trial. And one of the nice things about our proprietary construct design technology is that we can specifically look at the generation of synthetic designs for our antigens of interest, and that allows us to build in the ability to generate cross protection, cross neutralizing responses. Another nice dimension of DNA vaccines is that we have a huge amount of experience in our oncology programs for really safely re-administering our vaccine many multiple times. So for our GBM program, for instance, we've given our vaccine over 10 times to each of our subjects, and we see no impact from increased reactogenicity or no vector-induced issues there as well. So as we're kind of thinking ahead to COVID-19 as a disease, you know, quite realistically being an endemic situation now moving forward, you know, the ability to be able to re-administer vaccines is going to be absolutely key. We've also historically looked at both homologous and heterologous boosting platforms using DNA, and it's really very well suited to being that kind of companion option. So in a few slides, I'll talk a little bit more in detail about the safety and tolerability of our vaccines. And this is obviously in the context of COVID-19, but one of the really nice elements is that it completely mirrors every other pathogenic target that we've looked at too. So it's completely agnostic really of what is encoded, and that safety and tolerability profile is translated across the platform. We have a high patient uptake, and I'll talk a little bit about our delivery technology, which is probably pretty new to most of you, but really it has minimal side effects, and generally patients describe it as very similar to a standard flu shot. As I mentioned, one of the really nice aspects about our DNA vaccines is the reliance on the lack of reliance on frozen cold chain. So our vaccines are stored long-term or stockpiled long-term at 228 in standard refrigerated conditions, but that we have 1 year of stability at ambient temperatures, so anywhere between 19 and 25 degrees C. but also a month of stability at 37 degrees C. So as we're thinking, I mean, I think we've done a fantastic job in rollout in kind of developed countries, but as we're thinking about trying to ensure global access to COVID vaccines, the need for vaccines with a thermal stability profile that really can take away the complexities of needing frozen cold chain will be absolutely key. Of course, because our technology is based purely on a genetic sequence, the discovery or the design of our constructs can be done extremely rapidly. In the case of COVID-19, we received the sequence of the virus from the Chinese authorities, and just a matter of a few short hours later, we had our fully designed INO-4800 vaccine ready to go. And so that, I think, as we think about future pandemic outbreaks where, you know, we have a lot to learn from the situation we're in today, but certainly rapidity and speed is very key. Then, you know, that is a real upside of this technology. And of course, as Katie also mentioned, the need for manufacturing upscale is absolutely key in a situation like this. And that's certainly something that we've been very diligently working on over the last 12 to 15 months. So as I mentioned up front, we combine our highly optimized DNA constructs with our optimized delivery path platform technology. And this, I'm assuming, is something that most of you won't be too familiar with. So let me talk you through what we're discussing here. So what you're looking at on the slide is our Cellectra 3PSP delivery device. To give you an idea of scale, it's about the size of an electric toothbrush and about the same weight as well. This device delivers the DNA construct to the skin of the subject, so it's very, very minimally invasive. The needle electrode parts are only about 3 millimeters in length, so it really just penetrates the skin and no more. Historically, there's always been kind of this issue about DNA vaccines not being efficiently delivered into patient cells, and we really believe that our Cellectra technology is the solution or one of the solutions to that paradigm. And really, we've shown pretty extensively, both clinically and preclinically, that our delivery technology really incredibly enhances that uptake into the relevant cells of choice. And because, of course, this is intradermal delivery of a vaccine, what we're talking about here are keratinocytes primarily, but also APCs in the skin as well. The device itself really was in conception prior to COVID-19, which was incredibly lucky. We really had a mandate when thinking about our Lassa fever vaccine, for instance, to develop a delivery device that could be used in non-developed settings, clinical settings, so where access to electricity may be difficult, and something that would be really very simple to use without any extensive training. So the handset itself can be used 5,000 times. It uses rechargeable batteries, so something that could be easily charged off a solar panel. But each individual patient has a disposable array part which fits on the tip of that device. The whole administration process takes about 6 seconds, so not really terribly far off a standard administration with an IM vaccine. It's, as I've mentioned, an intradermal technology, so really the tolerability profile that we see is really just some mild AEs on the surface of skin. You generally get a little bit of redness on the surface of the skin that resolves within 28 to 48 hours. So very, very limited side effects, and we get very, very few systemic side effects. And from a patient experience, really the delivery with our device has really a very high subject tolerability. And generally our subjects rate it at about a 3 out of 10 on a VAS pain score, which is very, very similar to what you see for your annual flu shot. So as I mentioned, in addition to COVID, we have a very broad pipeline. My particular area is our infectious disease programs, where prior to COVID, We had an active program for MERS, and that was in partnership with CEPI. Also our Lassa fever vaccine, which was also in partnership with CEPI. And we were incredibly proud to be the first entity to get a Lassa fever vaccine into the clinic, and then also one into the clinic in Africa. So that's something that we're incredibly proud of. We also have active ongoing programs for our Ebola DNA program and also our HIV programs as well. But with a specific emphasis on COVID-19, we're extremely grateful to the support that we got from the Bill and Melinda Gates Foundation, CEPI, the US Department of Defense, and IVI and Advaccine, who are our collaborators in China and in Korea. I won't go into details on the rest of the pipeline. Suffice to say, we have a large HPV-associated portfolio, our immuno-oncology portfolio. And last, but by no means least, is our ability to use our DNA constructs to encode monoclonal antibodies. And that's something that's really incredibly exciting and is a potential option to bring the power of monoclonal antibodies to developing nations where just the cost at the moment is completely prohibitive. So INO-4800 is our foundational vaccine against COVID-19. It was designed back in January 2020, of course, against the original Wuhan strain. It's a 2-dose regime where we deliver vaccine to the skin at day 0 and at week 4. In addition to 4800, I'm not going to speak about it at great length here, but in addition to 4800, like all of the other developers, we're also looking at a next generation COVID vaccine, and it's going to be a pan-COVID approach, where we've really harnessed all of the accessibility to the available sequences on GISAID, of which I think there are now over 500,000 of them, and really use that to try to predict what we are potentially going to see in the future with variants of concern, but certainly for the known variants of concern as well. So that's an ongoing program. It's currently preclinical, but we're hoping to get that into the clinic this summer. So back to our foundational vaccine, INO-4800. Our phase 1 and 2 dosing regimes are complete. Those were both US-based trials and in adults 18 and over. And in both cases, the vaccine elicited a broad immune response, and I'll give you some examples of that. But we generated binding, neutralizing, and cellular responses at week 6 and onwards. And an interesting element of our phase 1 trial, which is particularly pertinent right now, was then we amended the trial so that we could come in and boost those subjects 6 months to 10 months later, post their first regime of vaccines. And that work is about to be published in the very near future. We're just wrapping up our phase 2 trial just now. The data is on BioRx at the moment, and we're hoping to start our phase 3 global trial imminently. And as I mentioned up front, we have ongoing clinical trials both in China with Advaccine and IVI in Korea. And as I mentioned up front, we've been working extremely hard to scale up our plasmid manufacture, really in a global footprint, to ensure that there are relevant numbers of vaccine available. So some of the really key differentiators of INO-4800 is the safety and tolerability profile. I'm going to give you some examples of that in the coming slides, but really we see that as a key differentiator. We don't get systemic fevers or malaise, anything like that. It's really predominantly just this redness at the injection treatment site. And as we think about the need for key personnel to be available post-vaccination, I think that's going to be very key in the future. From an immunogenic perspective, we were able to generate those favorable T cell responses, both CD8 and CD4, although predominantly CD8 responses, in addition, of course, to the antibody responses too. As I mentioned, something I think very important will be the thermostability profile of the vaccine. as well as the ability to repeat administrate these vaccines in the future, as it, I think, seems pretty obvious that we are going to require boosting vaccines. So this is just an outline of our phase 1 US trial. It's published in eClinical Medicine. This was initially looking at 40 subjects, looking at a 1 and 2 mg dose, and obviously taking the subsequent immunogenicity readout from that. We then subsequently expanded that trial to 120 subjects and looked at a range of different dosing options, as well as stratifying the age groups from 18 to 50, 51 to 64, and then 65 and older. And of those 120 subjects, 93 have now been re-administered a booster dose between 6 and 10 months post their second dose. So this is the safety and tolerability profile for our phase 2 data. So this is just shy of 300 subjects. Again, was a US-based trial. And on the left-hand side, you can see the profile after a single dose, and on the right-hand side after the second dose. And hopefully you can see that really the two profiles mirror each other very nicely. So we're not getting any inducement of additional reactogenicity the more doses that we give. It looks very much the same between 1, 2, and 3 doses of the vaccine. And generally, it's really just these local redness events is by far the most common AE that we're reporting. So this is some of the very early immunology data from our phase 2 trial. So you could see that in blue is our 1 mg dose of our vaccine, and in green is the 2 mg dose. All we're showing here is our week 6 data, and with obviously with the placebos as well here. So you can see that for binding antibodies, you can see a very clear dose response with the vaccine administration, and really the same can be said for our pseudovirus assay as well. And importantly, in the context of today's talk, you can see that we're generating some decent ELISpot data there as well. Oh, and one thing I should mention is that this profile was really seen in all age groups. So that's something that we're very, we feel very confident about, that we didn't see a drop off in immune profile with our older age group subjects. So that's something really very important, especially as we know that they are disproportionately, of course, impacted by COVID. So as it seemed completely appropriate, I think at the end of last year, we were all aware of the impact of the circulating variants. So it was very clear to us that we needed to ask the question, did our foundational vaccine INO-4800, which as I mentioned was based on the Wuhan strain of the virus, how does it face up against the new circulating strains? So as such, we performed several different immunological assays to ascertain the answer to that. And on the right-hand panel here, what you can see is that for our ELISpot readouts, that really there was zero impact on our T-cell responses against any of the variant strains. We looked at B117, B1351, and also the P1 strain as well. And you can see that there was no negative impact at all on those T-cell responses. So that gives us a lot of confidence in the ability of INO-4800 to really combat those circulating variant strains. And completely pertinent to today's discussion, of course, we are currently evaluating the circulating variant strains from India at the moment. On the pseudo-neutralization standpoint, again, we did the same assay using sera from our clinical subjects. And you can see there is a modest drop-off with B117, although we don't think that would impact the mechanism of protection of the vaccine. But interestingly, for the P1 Brazilian variant, we saw no impact on the neutralizing antibody responses. For B135, the South African variant, we did see a more pronounced drop-off. But again, we're ascertaining in challenge studies currently whether that actually would impact the ability of the vaccine to protect. So overall, we're really very emboldened, and we feel very pleased to see the profile that we've generated against the variants. So we're very confident for 4800. But as I mentioned up front, we're also working on a pan-COVID vaccine option at the moment, which we will be publishing in the near future. So this is my last slide, I think, really just talking through, coming back kind of as a summary of what our DNA medicines can do in this pandemic, and also really in future pandemics. So we really believe that DNA medicines have a role to play. They're very novel, but very efficacious, and that using kind of our AI technology allows us to design and develop very unique, bespoke vaccines. We have that broad immune response profile, which, you know, in the case of the variants is very important. And we've demonstrated protection for our other vaccine platforms in a wide range of animal models, and are just, you know, on the cusp of starting our phase 3 for COVID this summer. And again, you know, the boostability of the vaccine is going to be really very, very key as we think about the future of this pandemic. We have a very nice safety and tolerability profile, very nice thermostability profile. We've done a lot of work scaling up our global manufacture, and a bit out of the scope of today, but we also have those very clearly defined regulatory paths. And so with that, I'd like to thank all of our supporters, our collaborators, our funders, some of those that I've mentioned in the talk today: CEPI, the Gates Foundation, the US Department of Defense, have been absolutely instrumental, as has our collaboration with Public Health England and Cesaro in Australia. And lastly, of course, my heart goes out to our subjects in our clinical trials, without whom we wouldn't be here today. So thank you so much for listening, and more than happy to take any questions if we have time to do so.
Thank you very much indeed, Kate. Really appreciate you taking time to present the story of INO-4800. That's fantastic. We have time for one question from Uchol. He asks, is your vaccine-induced neutralizing antibody titer comparable to the titer in convalescent patients, or maybe comparable to the antibody titer elicited by mRNA vaccines?
Yeah, it's a great question. And certainly, I think on the convalescent, we're definitely within the convalescent panel, but probably on the lower end, frankly. And, you know, really the mRNA vaccines do an amazing job at generating very, very robust neutralizing antibody levels. So we are certainly not at their level. However, I would say, you know, it's kind of open for debate at the moment, what levels are actually required for protection. And certainly in some of our primate challenge work, we've had animals that have baseline neutralizing antibodies but are completely protected from challenge. So it'll be very interesting, kind of as the science proceeds on the correlates of protection, to see, you know, if those extremely high levels are required, or if something lower is protective, you know, given the input of the T cell responses.
Yeah, no, great, thank you. Right, well, thank you very much indeed.